Okay, so amplification of gene of interest using PCR. So PCR stands for polymerase chain reaction, right? Three step. First is denaturation, second is annealing, third is extinction. In denaturation, there is high temperature induces the separation of two templates of DNA. Okay, in annealing there are two sets of primers that are attached, and in extinction using the DNA polymerase and Taq polymerase plus deoxyribonucleotides. So extension of the primers. So this is third step, that is extension of the primers. So three steps and amplified into 1 billion times, that's it. And the temperature is high in denaturation, a little bit lower in annealing, and then a little bit raised in extension. So this is, and Taq polymerase is isolated from a bacterium, that is Thermus aquaticus, found in high temperature. Okay, and high temperature induced denaturation of double stranded DNA, already said you. Now two types of bioreactors for large amount of production of, if we need. So bioreactors, in there is like simple stir tank bioreactor and sparse stir tank bioreactor, right, through which sterile air bubbles are passed. Okay, so both the system have like agitator system like mixing, and this agitator system helps in mixing of like you see in written here, like um, the base is simple stir tank bioreactor has curved base. Okay, that facilitates mixing of the reactor content. Now the stir facilitates even mixing and oxygen availability throughout the bioreactor, that's it. Okay, now kind of both they have like agitator system and oxygen delivery system, foam control system, temperature control system, pH control system, sampling ports, so that small amounts of the culture can be withdrawn periodically. In diagram you see first one is motor, then below that foam baker, then flat bladed impeller, then culture broth, then sterile air. On left hand side acid bed for pH control and steam for sterilization. Now in second diagram, like sparse tear type bioreactor, the bubbles increase the surface area, right, increase surface area for oxygen transfer, that's it. Now downstream processing, like after the biosynthetic stage, so before everything it is biosynthetic phase. Now in downstream processing there is separation, then purification, then preservatives, that's it. So this is downstream processing. Now